Thanks everybody for coming. This is a great turnout for Eric. Um, Eric's assistant professor at USC in the Department of Biology. I'm sure students are here too. Thanks for coming. He is a plant pollinator guy. And as much as I know about his research is he's studying sand verbenas out west. And in this part of the world, he inherited a very historic moth collection, which is going to become part of this talk. So I'm glad you'll get to hear about that. Thank you for coming. I've heard this talk in a different iteration and it's fascinating. So I'm glad y'all are here. And thanks for your patience.
Awesome. Thanks for having me. If you can't hear me, just yell at me and I'll speak up. So today I'm going to talk about natural history in general. And by that, I mean sort of the study of organisms in their natural environment, whether they are plants or animals, native or non-native. And natural history is something that I think all of you guys do intuitively and think of as, a means to an end itself. However, it also has, sort of importance and I would say sort of the foundation of biology. We've learned a lot about how hearts work by studying small mammals that have crazy heart rates. We've been able to understand and to a large extent control tropical diseases by learning about mosquito life cycles and mating behavior, as well as sort of like the symbiotic bacteria that they interact with. And topically now, unfortunately, this is cut off here. I don't think I can make this go away. But oh, there it is. The like GLP-1 agonists, these weight loss drugs, the basic science that set up the sort of biochemistry was done on Gila monster venom. So, natural history leads to many sort of well down the line positive outcomes for people. Oops. And not just in medicine and human health, but also in our pollination. We now understand better how native pollinators help with crop pollination. In South Carolina, topically, we understand how preserving coastal vegetation helps us during storm surges and hurricanes. And natural history is also the fundamental knowledge behind conservation. We've had really good luck with certain things like sea turtles. We're doing okay with other things. We've done really poorly with others, unfortunately. And just knowing the sort of basic life history, the organisms that these organisms interact with, what these organisms are, where they're distributed, really forms the basis of that. Whoops. But, and this is something that teaching undergrads, they expect me to know everything about every plant and animal. Don't we know it all already? And if you read the news, you might get that sense. We have a pretty good knowledge of exactly to the individual how many right whales are in the whole North Atlantic Ocean. We know almost to the individual how many Florida panthers are in the Everglades and in Big Cypress. Every time a wolf crosses the Oregon-California border, there's a press release about it, which definitely leads us to believe, hey, we know everything. And that idea that we don't know about the natural world around us here in the United States has become kind of a joke. I mean, how many of you actually think that they're going to find Bigfoot on one of these TV shows one of these days? Obviously, they're not. We're not going to find a sea monster in Lake Champlain or Loch Ness. And so, okay, well, there's stuff elsewhere in the world. It's probably in far-flung locations, in the deep forests of Madagascar or the Amazon or Southeast Asia, or perhaps deep in the ocean, where all these crazy organisms live. And for plants, topically, we think that due to sort of species accumulation curves, we think that the vast majority of plants that we have not described yet, that we have not put a name to, are probably in tropical areas of South America and Africa. So there's this sense that, oh, well, we figured out everything here. We've got to go elsewhere. Um, you know, Perhaps there's a sense, maybe not amongst you, but there's a sense that we know pretty much all that we can about South Carolina animals and plants. And I ask my students this question at the start of my entomology course every year, like, what is the likelihood of discovering an insect species in South Carolina? And about half of them say, oh, that's probably pretty unlikely. And half of them say, well, maybe there's something out there. We discover new species every year. This is a wasp moth we just published that I found a couple years ago that no one had ever found before. And even if you're not likely to discover a new species, we're out there all the time and we don't find them that often. How likely are you to add a species to the state list? Well, I can tell you from looking on iNaturalist, which I'll get to, someone found a tropical sphinx moth in their garden in Charleston. They've even got their gardening gloves on in the picture recently, and that had never been seen in South Carolina before. So that's also possible. But I would argue that, we get a little too much caught up in the, say, trading card aspect of this, finding new species. And I would say that the currency, what we're really interested in, shouldn't necessarily be just, ticking off species. So I'll talk a little bit at the end of the talk about moths, since most of my South Carolina research is on moths. And we, go out, we put out black lights, we collect moths as adults. The adult stage of a moth is really beautiful. It's like, it's wonderful and diverse and all. But really, this is not the stage where moths matter. A lot of moths, this guy included, they don't feed as adults. They live for 4 or 5 days. They mate, they lay their eggs, they die. Most of the action that a moth does, and most of the time that it's like really important for the ecosystem, is when it's a caterpillar and feeding on plants. You guys probably are less fans of them than I am. But they're feeding on plants, so they're moving energy up the trophic chain. These are hugely important food for birds. The estimate for a brood of chickadees is that the parents have to feed them around 6,000 caterpillars in the 3 weeks before they fledge. They're also really important food for wasps, spiders, lizards, etc. And they're also very cool in their own right. But this isn't what people look for when they describe a moth. So there's thousands of moths in South Carolina that we know very well from their adults that we have no idea what they're doing. We don't know what plants they rely on. We don't know what eats them. We don't really know their place in the community. And in fact, in South Carolina, this is one of our most common butterflies in the butterfly censuses that I used to do in Charleston County. This would be either the most or the second most common species, the red-banded hairstreak. It's around Columbia. People know that the caterpillars live in the leaf litter, which is a weird thing for caterpillars to do, but we don't really know what they're doing down there. We don't know if they're specializing on one type of dead leaves. We don't know if they're even eating dead leaves at all. They could be eating things like fungus, or there's another species in this group called the harvester here that eats aphids. So it's possible that this is actually a predatory caterpillar living in the leaf litter. No one knows. And you can see hundreds of these on a daily basis if you look. So there's a lot more to know about even sort of, charismatic species, let alone all of these tens of thousands of tiny little species of bugs that surround you every day. Tiny little flies, solitary bees, moths that have a wingspan of 2 or 3 millimeters, let alone beetles, which are the most diverse animals on Earth. And the vast majority of these probably don't even have names. Haven't even put a name on them, let alone determined what role they play in a community. And the same goes for plants. So we can make what we think of as decently good guesses about what pollinates a plant due to floral morphology. Big tubular flower that's red, probably hummingbird pollinated. Big open flower, possibly bee pollinated. However, most species here, no one has really formally done the pollination biology of, And even in those species like common milkweed, where people have studied them over a large range, they've often found that in different areas, they have different pollinators. So even for species here that we do have some information, there's no telling if when that study that did determine the pollination biology in New York, whether that same, those same relationships are occurring here in South Carolina. And obviously, knowing about pollinators is really important for preserving or conserving plants, and knowing what else they rely on and they feed in any community. I would also argue that collecting specimens, and John does this obsessively, and documenting records, and I'll get back to that part, is hugely important. And I would say that natural history specimens form the basis of most of what we know about distributions and taxonomy of organisms, and they often get used in really creative ways. This is a paper that came out last summer at some point, and what they were interested in is they were interested in dating when plastic pollution started, and I think this was in the Netherlands. And so they went back to these caddisfly casings. So caddisflies are these aquatic insects that the larvae sort of ball up a bunch of pebbles and they live inside it. And taxonomists collect these. They put the casings in a museum. They sit there, and they went back to casings and they pulled out microplastics or what might be microplastics, and then analyzed them chemically. And they found that the first instances of microplastics in what they termed as pristine springs in the Netherlands were found in 1971. The term microplastics was not coined until 40 years after that. And the taxonomists that were collecting these things surely had no idea that they would be useful in tracking when this pervasive pollutant started. The same goes for herbarium sheets. There's a whole sort of sub-literature on tracking pollutants, especially heavy metal pollutants from industrial, like during the Industrial Revolution, on herbarium sheets. So you can take a swab of the leaf tissue or a swab, and analyze it for heavy metals and track pollution over time. And really, these specimens, as well as field notebooks, site records, also photographs, are really our only links to what past communities looked like. We, unfortunately don't have time machines yet and can't go back and figure it out. So if we're going to determine anything about change in the world, we, at least in biology, we need to have those records, preserve them, and use them in creative ways. And I'll say that obviously, 40 years ago, people didn't, think of taking plastics out of specimens, or even, looking at pollution on specimens. And I have no idea what these moths will be used a century from now, but I can guarantee that there will be new technology and new creative uses of them. And we'll come back to that at the end of the talk. And next, I would argue that natural history has never been as important as it is today. So invertebrates, we know that species are going extinct at an unprecedented rate. We only know this for vertebrates because we have really good records of birds and mammals, because people care more about birds and mammals than they do about plants and insects. However, the vast majority of evidence suggests that insect diversity and abundance is also declining. And I'm sure you've seen these scaremongering headlines over the last few years. However, natural history is something that is not taught in schools as much as it used to be, is not something that a huge amount of students really care about. The undergrads and grad students that are here accepted. And various people have sounded the alarm for this, that natural history has been displaced, expelled from the ivory tower, and is presently seen as less prestigious than other disciplines. Paul Dayton is a really famous marine biologist. He basically discovered how much life there is under the Antarctic ice sheets. And that was just basic natural history when he was doing it in the 70s. And Josh Tewksbury used to be head of science at the Nature Conservancy. He decided to put some numbers to this. So he collated the number of herbaria over time and found that they peaked in the 1990s and they've been declining ever since. I interviewed at the University of Georgia, and I talked to the herbarium curator there, and she said every year they get offered small college collections, and in some cases she can't take them. So those are people's lifetimes of work, sheets that are just tossed in the garbage. Recently, you may have seen this, it was in the Times and the Washington Post. Duke, which is the 7th largest herbarium in the country, I think, somewhere around there, decided that the herbarium was not bringing in enough money to cover its costs and that the administration didn't want anything to do with it anymore. And I don't know what the current state of this is, but it's closing in some shape. Additionally, the number of people that are getting PhDs in natural history disciplines has been declining pretty steadily. And this is, of course, the people that can teach the next generation, which is problematic. And then most problematic is that if you were a biology student in the 50s, in order to get a major in biology, you had to take 2 or 3 natural history courses. Now at USC, as well as almost every other school, you can get a biology degree without having taken a single natural history course, which is problematic and troubling. So there's a lot more to discover. Hopefully I've convinced you that it's important. Why aren't we all studying it? Well, this is sort of more abstract and philosophical. And I think that really the decline in natural history is twofold. I think that biomedical sciences are seen as more important. We all benefit from biomedical sciences. That's pretty important. I would argue that increased investment in that side of biology shouldn't come at the expense of natural history. Instead, we should be increasing both. The other I think, more fundamental problem is that people see this dichotomy in science, that there can be descriptive science and observational science, and then there's this hypothesis-driven experimental science, and people think that this science is much more important. However, I would say that this is a false dichotomy. And what I mean specifically, and this comes from Casey Dunn, who's actually who does genomics of weird sea creatures, but that there's sort of two axes that are orthogonal here. You can do hypothesis generating science, you can do hypothesis testing science, you can do descriptive and observational science, and you can do experimental science, and that hypothesis generating descriptive science is not across from hypothesis testing. And if it were, we would never know anything about the solar system or the atmosphere, right? So there's a lot of fields that are almost entirely descriptive and observational, and that doesn't mean you can't come up with interesting hypothesis tests. And of course, it's really important to generate hypotheses before you test them. So if we hadn't spent a long time breeding crops, we wouldn't understand basic genetics. There was crop breeding that had gone on a long time before Mendel noticed inheritance and figured out the basic, the basic patterns of at least of Mendelian genetics. So I think to some extent, natural history has been sort of denigrated because it happens to fall largely in this quadrant. However, the things that come out of it certainly span the full range. And I would say that in general, natural history as an academic discipline is dead, with some exceptions. And this isn't only a problem over at USC and at college campuses. There's this beautiful book from about 15 years ago that talks about how children are spending less time outside and that this leads to worse well, people are spending outside less time outside, especially children, and this leads to worse educational outcomes, and things like attention span, focus, are positively impacted by time outside. So I hope I've convinced you that natural history is really the fundamental knowledge that we need to conserve species. How many of you know this bird? Bachman's warbler, the first one. The type specimen was shot in downtown Charleston. One of the last times it was ever seen was at Congaree, and it went extinct about 50 years ago. Um, and hopefully, I've also convinced you that doing natural history and knowing about the organisms and communities around us are good for us as well. However, can you guys see the bottom of the screen? Uh, That's better. Um, however, as I also told you, there's not a whole lot of training, funding, and support for natural history study. So what are we supposed to do? Okay, that was all pretty pessimistic. I hope the second half of the talk is optimistic. But let's go back to natural history in say the mid 1800s. So let's say that you guys, as interested plant people, you don't have field guides, you don't have an herbarium here yet. What are you supposed to do if you collect a plant down in South Carolina or say up in one of the soon-to-be states? Well, what do? You have to send it back to someone in, at Harvard, at Yale, in New York. They might put a name on it. They might, file it away. You probably won't know a whole lot. There were obviously some books, but this was how it largely worked. And the folks that were collectors were generally not the ones that were describing the species. They were not the ones getting the credit for it. And so a lot of field biologists, especially field biologists out west, got pretty sick of this. And the first scientific journal that was founded west of the Mississippi was out of the California Academy of Sciences, where Catherine Brandegee, who started the journal, was the curator of the herbarium. And so she basically started a field journal. There were descriptions of species. There were, descriptions of songs of birds, California parasites, whatever the sort of Cal Academy crew was interested in publishing. This gave them an outlet that wasn't run through one of those major institutions. It's also hilarious to read. This journal only lasted a few years, but it's hilarious to read it because these people like Marcus Jones, who was a really talented field botanist, had these like long articles where they had little whole paragraphs ranting where, it doesn't express our feelings when we see a new monograph from men who would not know their new species if they saw them alive. It's worth looking up. The Biodiversity Heritage Library scanned all of the issues in. You can read them. So I think we're doing better than we were in the 1890s. But obviously, I told you some sobering things a minute ago. So what's happening now? Who's making the discoveries that are being made? So let's go back to Catherine Brandegee. So her protege was Alice Eastwood, who took over from her at Cal Academy. She's got a bunch of both genre and species named after her. And in the late 1800s, she found a new species of manzanita on one of the hills in San Francisco, I think like concentrated around some cemetery, named it Arctostaphylos franciscana. And, this sat there. It was up on the cemetery for quite a while. And then San Francisco went from a really small city to an enormous metropolis. And unfortunately, that species seemed to have gone away with it until October 16th in 2009, when Dan Glusenkamp, who was president of the California Native Plant Society, was stuck in traffic in the Presidio. And he was stuck in traffic because they were doing a bunch of road construction. And he noticed in this clover leaf that was about to be demolished a prostrate manzanita. And so the next day he called some of his botanist friends. They jumped across traffic and they found out that this was the missing manzanita, the last individual of it. And Caltrans uprooted the whole thing, moved it to a secret location. There's now cuttings of it in a bunch of botanic gardens in California. And my advice from that is that like people drove within 15 feet of that thing for like 70 years and never noticed it. And if that can happen when there's that many people that close to that plant, keep your eyes open. I have a more personal story. You guys all know white clover. It's on every lawn in campus. It's on every one of your backyards. One day I noticed that there were these little blobs on it. You can't see it well on here, but there's actually a caterpillar sitting inside each blob. So these are leaf mining caterpillars. The epidermal layers, so the outside of the leaves, are not all that nutritious. But if you're a small enough insect living inside the leaf and being able to eat all the good stuff is a really nice way to be. And I figured, oh man, I would have noticed this if because white clover is everywhere. So I picked some of these and I sent them to a friend, Charlie, who was working on leaf miners. And it turns out that this was the first time it had been collected in North America. It was actually a South American species. And in fact, within a few months of finding it, Charlie started soliciting, like telling people to go look for it. It turns out that it was already from California to the Carolinas, and just nobody had noticed. And so when Charlie set out to write the book of the leaf miners in North America, which is not a real physical book, it's an ebook. It's 1,800 pages long right now. He's discovered at least 16 species in his tiny yard in Massachusetts. Two other species that had not been found in North America. And he's described, and I think this is long out of date, over 400 species. And this is just from looking carefully at the organisms that live inside leaves and leave little tracks or little blotches on them. So, okay, I also wouldn't have known that manzanita, and I had to send those leaves to Charlie to figure out what it was. However, we live in an age where you can get knowledge. You don't have to send your specimens to Asa Gray at Harvard and have him put a name on them. When was the first field guide invented? Now there's a field guide for everything. There's this beautiful new field guide for butterflies of the Carolinas. You can get field guides for hoverflies and grasshoppers. How old is the field guide? 30s, actually. 1934 is when Roger Tory Peterson published the Guide to Eastern Birds. And they really didn't, I mean, there were some bird guides. There were the golden guides, those really tiny ones, published in the 50s and 60s. But it wasn't really until the 70s that Peterson Guide started to put out, stuff to a bunch of other groups. And so this is a relatively modern invention. People 100 years ago could not have imagined that you had this full color, beautifully illustrated guide to all the grasshoppers of the U.S. The flora of North America is a set of really dense taxonomic keys that takes up how many shelves in an herbarium? A whole bunch. Big, dense taxonomic works. You can pull it up on your phone now and get the keys and the species descriptions right there. That's amazing. Leaf miners, the same thing. It's 1,800 page PDF, keys to every leaf miner in the US. You can just pull it up on your phone. And there's a whole lot more. Biodiversity Heritage Library, unfortunately, has been cut by this administration, but it's still up for the time being. And it's basically the Smithsonian has scanned in 63 million pages of basically old natural history books. And then every taxa, if you're interested in birds, Cornell has a wonderful website. If you're interested in moths, Mississippi State, and so on and so forth. And these are like just truly spectacular resources. And many of them are made by interested amateurs. Jeff isn't really an amateur, but To some extent he is, and he's made these beautiful guides for for North Carolina butterflies and dragonflies that I refer to all the time. This has been coupled with this rapid rise of citizen or community science. The two biggest platforms for that are eBird and iNaturalist, both of which you can get as apps on your phone or log into on your computer. eBird is the longest running. I've been using this since 2006, and I think it was started about 2003. And here's just the records of chickadees georeferenced around Columbia. And these are just a few of the over 2 billion bird observations that have been recorded. And basically what you do is you walk around, you say how far you walked, how long it took, so that there's some effort data, because obviously you're going to find more birds if you've been out for 2 hours than 10 minutes. And then you say how many of what species you find. And this data goes into a database, and it allows people to track populations over time, both through different years as well as seasonally. So they've made these beautiful illustrations of bird migration. This is my favorite bird, the scissor-tailed flycatcher. It breeds in the southern Great Plains, and then it migrates back to its wintering grounds in Central America. And so all of this data that was able to make this really granular, wonderful map was contributed by people like you who were just out birding. And this has permitted us to really understand where they are at any given point in time. And over time, and eBird's been running for more than 20 years, we can document range shifts and population declines and increases. The other big one is iNaturalist, and here is all of the records of everything around Columbia. You just kind of take out your phone, take a picture, it uploads it to this database. You can put an ID on it. Other people can put an ID on it. It's not quite as good because you don't know if you took a picture of one monarch and there was only one there, or there were 100 there. But still, that occurrence record is just as good in some ways as a specimen in a box. There was this species at this location at this time, and those photos have been used for all sorts of cool studies as well. So here's what they look like. I collected this moth in 2023. Turned out to be a first state record. It's up there so other people know that it's there. We also have technology that, besides those internet resources, that people couldn't have dreamed of. Microscopes used to be extremely expensive. You can buy a microscope that takes pretty decent pictures for $20 on Amazon that plugs into your computer or Bluetooths into your phone. Same goes for binoculars. Binoculars used to be really expensive. For $100 now, you can get a really good pair of binoculars. So I would say that we live in really a golden era of natural history resources that was truly unimaginable 100 years ago. And you don't need, these sort of ivory tower gatekeepers to do good natural history and really to contribute meaningful data to science. And so I want to highlight a couple people who have made really big contributions. These little moths, Eupithecia, are in some ways the bane of my existence. There's 300 species. They're all about this big. They're everywhere. No matter when you put up your traps, no matter where you are, you get these things. They all look the same, sort of. There's some are slightly browner, some have more lines. They mostly come out looking like that, where they're all worn off. They're just terrible, terrible moths. And the only way to identify them is to dissect out their genitalia, because genitalia are really good identification characteristics because, two species that have differently shaped genitalia can't mate. And you can imagine how big the genitalia are in the last segment of the moth. You can imagine a moth that's this big, how big that is. So needless to say, this is a really, really hard thing. And I have drawers of Eupithecia that I have not and will not dissect. and enter and know everyone. They're sort of like the bane of most lepidopterists' existence. And a few years ago, JoAnne Russo, who was a basket maker, went to art school, was a basket maker of enough renown that she has pieces that are archived in the Smithsonian. And when she retired, she figured she needed a hobby. She went to a moth course and she got really into these genitalic dissections. And for some reason, Eupithecia, these boring little gray moths, really caught her attention. And so she spent her retirement for about the last 10 years making these wonderful identification resources for Eupithecia. And she's an artist. Yet, every lepidopterist I know sends their Eupithecia to JoAnne because she's happy to ID them. She wants specimens from everywhere. And she's really the only person that can be trusted with these IDs. So she's really, and I send her mine as well. So she has really like transformed herself into the world expert on one of the hardest groups of moths there is. And then the last thing I want to talk about is an amateur setting the stage for the research that we're doing. So Richard Dominik was an ophthalmologist in New York City. And when he retired, he bought an explantation on the south bank of the Santee River. So if you're going down 17 from Georgetown to Charleston, there's those two bridges, and he bought a property right along the river on the south side. And Dominik, in addition to being an avid hunter and an ophthalmologist, was also a moth nut. And this is what he really cared about. And so pretty much immediately upon retiring and buying this property, he built a laboratory just to build a collection of moths. He converted a small cabin on the property into a moth trap. And by that, I mean, he put a whole bunch of lights in the center of it, and he got someone to build these automated shutters that opened at dusk so that the moths could fly in attracted to the light, closed at dawn, and then he could go in every morning and take whatever specimens he wanted. He also hired a full-time assistant, and he hired students from various universities to come over summer vacation and just prepare moths. So this was like possibly the most active like moth research place just about anywhere, certainly anywhere in the Southeast. And he was really into not just like documenting the moths, but figuring out the life histories. In fact, there's a few species that he's the first person to describe the life history, to find the caterpillars, to find out what they ate. So the cypress sphinx is one that he didn't discover the species, but he was the first one to rear the caterpillars. And he also documented, in addition to their whole life history, he documented interactions. So these are parasitoid wasp cocoons that he's mounted on, or that are naturally on this caterpillar, and he saved the specimen of. He sent his parasitoids to the University of Florida for ID, and those, of course, had which moth species they came out of, and all of this other valuable information with them. So his dad was president of the New York Stock Exchange. He retired at age 45, which I think tells you something about his financial situation. And one of the things he really dreamed about was getting a guide to moths. So there was one book about moths written by William Holland in 1898, and it actually has photos of various moths, a few hundred of them. But they're, of course, 1898 photos. There's not a whole lot you can really divine from them. So he hired folks from Yale, from the American Museum, from the Smithsonian, from the Canadian Museum, both to come down and collect and to curate his collection, as well as to work on sort of I guess this was inspired by the flora of North America, the moths of North America. So these dense taxonomic treatments of all of the groups of moths. At the same time, he also realized the value of identification resources for the public. So he hired and put up the money for Charlie Covell, who was a professor at the University of Kentucky, to publish the first guide to moths when the Peterson Guide started to expand beyond birds. This book was first published in 1977. It's still in print. It hasn't been updated. In 1976, he died. His collection ended up here in Zumwalt in around 1982. We don't know the full sort of early history of it, but it was sitting in an office in Zumwalt. That's next. So during those basically near nightly collections, he discovered around 26 species new to science. Some of these are like very enigmatic. This is known from one site in South Carolina, one site in Mississippi. This is known from a couple sites in the Southeast. This one, he found hundreds of. We have 130 of them in our collection. One was caught in 1983 in North Carolina. It hasn't been seen since. It may be extinct. And a whole bunch of others, some of which still don't have names. And so he left about 30,000 specimens here. We think there's around 30,000 specimens in the Smithsonian. They have 32 million specimens. They have not databased. We can't just look up what's there. We've gone through the drawers. We found whole drawers of his material. But he wanted a full collection of all the species left here in South Carolina. And so when I arrived here in 2022, I somehow inherited it and realized that this was a wonderful opportunity to do a resurvey. Nobody, like truly nobody, did that intense collecting. So we don't really have any idea of what a full community was like anywhere else in the past. So this was his property. Unfortunately, it's still private, but we're able to collect here, here, and here. And it's basically continuous marshland here and pine forest and Atlantic forest. And so over the past 2.5 years or so, we've databased around 100,000 moths. And while this is hard to believe, we're actually nowhere near done. Every time we go out there, we continue to find new species. And basically, if we're going to census the whole community, we need to stop finding new species. That's kind of our goal, to say we've gotten everything. So we've documented a huge number. I think we're up to around 45 or 50 species that Dominik did not get. The vast majority of these are northern expansions. So there's species that were known from Florida, known from Georgia, and that have marched northwards. And we think that this is largely due to the wintertime low temperatures getting much warmer in the intervening time. While Dominik was sampling in 1968, the Charleston airport had 68 nights of frost. In 2023, they had 8. And the average nights of frost while he was sampling was around 40, and the average nights of frost right now are around 20 there. So a lot of these species probably just couldn't hack it at that point, but it's warm enough now. There's also others that are invasives. We've documented this year alone 3 new invasives for the state. Hopefully none of them cause big problems. And then other things like tropical strays. And in addition to these community changes, we're also interested in how the actual species have changed. There's a few papers recently that have indicated that in a warming world, ectotherms, so cold-blooded animals, are probably spending more of their energy on metabolism. And so something like a moth that's a caterpillar for a while has to spend more energy, is going to come out as a smaller adult. This seems to be true in bees in a few studies. No one had looked at it in moths. I was convinced that, there's like so many things that determine size. The quality of the food, the timing of reproduction, all this stuff. I was pretty convinced that this was not going to be true. And I tried to talk Allie, one of our undergrads, out of doing it, out of actually looking at this. However, with the help of a visiting grad student, Vincent, who wrote this AI program to take images of the specimens and automatically measure them, Allie was able to compare thousands, truly thousands of tiger moths from the historic collection and the modern collection. And sure enough, she found that they are shrinking. And they're actually shrinking by about 6 or 7% in our biggest species, which is almost perfectly what we'd expect from metabolic theory, which was totally mind blowing to me. And hopefully, she told me actually this morning that she's working on getting the paper ready for publication. The other thing she did, and something that I really can't thank Dominik enough for, was that he pinned into the collection these little notes about when the species flew. So what months he would catch the species in. And this is really granular because, of course, March 31st is the same as March 1st here. And so, again, I didn't really expect that we'd be able to find any sort of shifting timing of flight seasons. But sure enough, most spring species are coming out earlier, and most fall species are coming out later. And most flight seasons overall are getting longer, partially because of that, and partially because some of these species are able to go through more generations a year. So even with that coarse data, we're able to document shifts in flight seasons. And, I was thinking about this when I was making this talk, and these caddisflies that are plastics, and I'm pretty sure that Dominik was not collecting those specimens to have them measured by a computer 50 years later. But of course, this has showed us something. And this is really one of the few examples of what we expect is probably going on in most insects worldwide. So to conclude, I'd say that in this day and age, natural history has never been more important. However, support for natural history study. So I'm not teaching as many natural history courses as I would have been 50 years ago, as well as support for natural history collections is also waning rapidly. There was this horrible example at one of the University of Louisiana campuses where they wanted a new football stadium, and they threatened they would get rid of this million fish collection. They would just throw it all away if they couldn't get it out in like some crazy short amount of time. They did, fortunately. However, it's really a wonderful time for natural history study. You can contribute data that is useful now and useful in the future. And a lot of the people doing it, definitely the majority of the people contributing data are amateurs like all of you. So keep your eyes out and keep your curiosity high. And I also sent these little leaves of this weird desert plant in California to Charlie because they had mines in them. And it turned out to be this cute little fly, which he named after me. So maybe something like that could happen. So I'm happy to take any questions. Come up and look at the moths. I hope I didn't take too much time. Great. Yeah. Oh, that's a really good question. Nowadays, you generally have to do at least a little bit of DNA sequencing. There's like this one section of one gene that we use for barcoding. So morphologically, sometimes two moths look entirely identical except for genitalic shape, and sometimes they look totally different. So this one, this one looks nothing like, I mean, it's only this big, but it looks nothing like any other moth. He knew it was a new species. We know it's a new species. There aren't enough moth taxonomists to actually put a name on it right now because they do have to like describe the genitalia, get it sequenced, etc. Is that roughly what you were asking? No, it's throughout the natural world. Yeah, I mean, taxonomy is a really difficult field because there is no one criteria. and you're trying to basically determine, is this thing, you know, probably not reproducing with these other things such that it's probably its own entity. And so morphology, life history, is it flying in the spring, whereas this other one's flying in the fall, and therefore there's, no chance of them reproducing with each other, as well as genetics as another line of evidence. So it's a whole bunch of them. And I mean, I found that wasp moth that I mentioned, but I did not do any of the formal description. I couldn't figure out what it was, so I sent it to an expert who was really excited because it was something that he hadn't found. But it took him 2 years to put together the evidence that it was a new species. Yeah.
Well, eBird, have you heard of Merlin?
Yeah. Yeah. And so Merlin is this wonderful, like it's almost magic thing where basically it records it, like, you hold up your phone and it tells you what birds are calling. And I don't know how eBird deals with that, if that stuff goes into the database. Do you know, Quaid? Yeah. But basically, and this is again largely amateurs contributed enough, like verified, this is what a hermit thrush sounds like, this is what a Carolina chickadee sounds like, in order to train this model. And it's almost 100% accurate.
There's been updates where it's gotten pretty bad, but
it's unbelievable. You should all check that out. It's like magic. Yeah.
It's a little bit like you kind of given up on natural history, regaining a position of importance, which I don't blame you, obviously.
Yeah.
Do you have, I mean, it seems like if we could all do a better job of linking natural history to economic outcomes, that perhaps that would be helpful, but is it too far gone for even that?
This is a good question. My impression is that a lot of academic research is driven by funding. Not my impression. A lot of academic research is driven by funding. And I was the last. NSF used to have a postdoc fellowship in natural history collections, and I got it in 2017, and that was the last year they offered it. And they do still have a systematics and biodiversity call, but the amount of money is really low. They fund a few proposals a year. So I think it would have to be a top-down, huge effort in order for it to regain that place in academia. But there are more people bird watching than ever before. This is a good thing that came out of the pandemic. There are more people involved in native plant societies. So I don't know. I mean, as an academic research discipline, we're all able to do it, but I couch it in evolution and ecology, and other people couch it in physiology and all.
I guess one of the things that prompts the question for me is I have an acquaintance, a friend who went to a venom conference recently, which was very cool, and I was very jealous that I never got to go. And as an aside, she keeps the stud book for cobras, and she got to go to stud school. Again, I never got to go to a conference. Anyway, I'm assuming that this had to do with the toxicity of the actual venom of the snake. No, it had to do with the economic benefits of what people were deriving from venom. And as far away as Australia there. And it was just a huge big deal, but who knows? Who knew? And so I'm wondering if there's more of that kind of stuff that's kind of below the surface that we don't realize, that maybe we'll circle back around and get to the NSF people who will then say, oh gosh, we should be paying more attention to this.
well, you brought up the Gila monster. If anyone, I'm not wishing this on anyone, so if you have a stroke and they get you to the hospital and they can get this clot buster into you in 30 minutes, you can make a 100% recovery, and that clot buster is based on the venoms from basically
Rattlesnakes.
So what do you, we make that link between the outcome that we all want, which is the useful medicine, and funding for academics to keep on studying the critters that might provide that? How do we make that link?
I'd say that we can't make that necessarily. And there's got to be more reasons for us to be interested in that than some kind of utilitarian aspect of their worthiness of life.
I say that's an idealistic view, and I wish it were true. But I'm a herpetologist. Every time people came to me about snakes, the question was, well, what good are they? And what they really mean is, what good are they to me? They want that the existence of that animal justified by what value it has to humanity. And that's an anthropocentric view, and we all recognize that, but we're dealing with human beings. So I think the more we can play this game along. I mean, if snake venom saved your life, you know, it might be something okay.
And when I was in graduate school, a long time ago, they used to talk about how wonderful it was that we had, you know, about this wonderful diversity, especially tropical plant species. And the big deal was that there's this periwinkle, Madagascar periwinkle, that could irritable. There's a one plant that we know of that was useful, that had been a model for some kind of lab derived anti-cancer drugs.
Well, the U in California,
Right.
source of Taxol.
And then there's digital.
Digital.
I mean, we talk about this, but there's a handful of these things. It's not like basket.
There's a handful of them. In some ways, because we really haven't explored. Sorry,
Yeah, no, one of my dad's best friends is a pharmaceutical chemist, and he thinks the era of like biological drug discovery is over. Because we now have good enough models of like what receptor, and they could just make like the compounds that potentially could bind to that receptor or whatever. And so he thinks that going out and looking for them is definitely already on the wane and is probably virtually over, which to me, like, I think I agree with John here that, like, I think tying it to human benefit negates the basic science, like, of an organism that we truly know nothing about. How do we defend studying it? Because we have no idea what the future utility of it would be. And, only 1 out of 10,000 of those things, is going to have some real tangible economic value to humans. Hopefully, some of those a huge value to humans. But that idea of tying basic science to economics and human outcomes certainly makes me uncomfortable.
But it's not you that's doing it.
That's true. Yeah, for sure.
And then my point was that's what people expect.
Yeah.
So, sometimes you've got to give people some of what they want to give them what they need.
Yeah.
But this is one reason why things like iNaturalists and the tools that are being made for the ordinary enthusiast to use and go out there and produce data that can be reinforced because of multiple observations from the community that's going to be so that when the scientist comes along who sees the potential for medicine or something like that for one species, and if that's not working quite right, so maybe something that's closely related to it might. So what would that be? How would I find out what that is? So the community can come in as another piece of the puzzle. And providing the data. Tools help us make sure that these data are accurate.
Sierra's gotten tons of seeds of our various amberbenas, and we've gone to so many iNaturalist points to find populations of it. I mean, it is really a wonderful community that we do lean on really heavily.
It's fun.
It's also a lot of fun. And it's fun to see people out there like taking pictures of things, and it's like, oh, I know what you're doing. Cool. Well, thanks for having me. Come check out the moths.